This is the second video for the kidneys, and we left off at arterioles. And let's see where these guys come from. First, I'm going to draw my hemisected kidney again. The ureter comes in, then becomes these, forks in the road called calyces. Come these forks in the road. There is my ureter. So at the end of these little paths, we have these renal pyramids. Okay. From before, we know that the renal artery brings blood to the kidney. So there is my renal artery. We also know that we have the, renal vein that takes blood out of the kidney. So renal artery this way, renal vein this way. Once inside, this starts to branch off. So part of it will go towards the bottom half of the kidney, part of it will go towards the top half of the kidney. So this will go this way, and we're going to focus on this left one here. So again, this would be the renal artery. Once again, this is going to branch off to get even smaller again. And it's going to go through the renal columns. It's going to go through the renal columns. Once again, this is going to branch off even further, becoming even smaller. And then all we are left with are these thin arteries that we call arterioles. So renal artery gets smaller and smaller. These subdivisions have names. We don't have to worry about that for now. Becomes arterioles. And you can consider arterioles as, very small, thin arteries. And since they are arteries, they still carry oxygenated blood. So they still carry oxygenated blood. Okay, but this is where the nephron, the structural unit of the kidney, begins. So this is where we can, this is where the nephron begins. Okay. Last thing to note is, let's use a different color. An area where tubes come out of a structure doesn't have to be the kidney necessarily. So this area here, we call this a hilum. Just an area where tubes exit or enter a structure. Okay, now on to the nephron. And let's try to get a bit of perspective here. Just going to draw my renal pyramid. So this is just a renal pyramid. This is in the medulla. The medulla, and above that pyramid, this is cortex, cortical tissue. Okay, so the arteriole travels up like so, travels up, and some of it, turns. We'll have a little folded blood vessel tissue like so. And now we can start to see the beginnings of our, let me erase this, make it a little bit higher, of our nephron. So there would be our folded blood vessel, and we'll have, a structure like this. And this is going to dip down into the medulla, back up, squiggly again, and then eventually go back down. But all this will be discussed in a sec. But this is where we are right now. This is the nephron, which consists of a few different subsections. Okay, so let's get started. So here is my arteriole, that thin blood vessel. And now we're going to have this, this different blood vessel, which is still oxygenated blood, but it is very thin and folded on itself like so, creating kind of this, globular shape. So let me label this. So this is still an arteriole. This is still an arteriole. But since this one is going towards this little, this ball of blood vessels, we call it afferent. And afferent means going towards. So similarly, now this part is also an arteriole, but we call this an efferent arteriole because it's going away from this, area. So this tissue right here is a little bit different than what we just mentioned, the arterioles. These are known as, well, this structure here is called a glomerulus. And these are known as fenestrated capillaries. Fenestrated capillaries. And let's see where this name comes from. What does it mean? So fenestrated means it has, many slits, many windows, many slits. And this is where filtrate, things that are going to the kidney, can escape through these little slits. So some will escape there, some will escape like that, some will escape, we'll have some escaping there. So how do we capture this filtrate? And that's going to be the next part of our nephron. So fenestrated capillaries has many slits. This allows filtrate to escape. Allows filtrate, things that we want to keep, to escape. Okay, now how do we capture this? So let's finish this down there. So here is my arteriole. Here is that glomerulus. Here's the efferent arteriole. And we have, again, filtrate escaping. Remember, this is a 3D structure. So coming out of the page, exiting the page. Now we have this next structure called Bowman's capsule. That's going to capture this filtrate for us. Capture this filtrate for us. So nice. Now we have reabsorbed. Now we can take in things that we want to keep in our body. And I'll list a few examples. We can take in things like glucose, and we can take small things like amino acids. Whole proteins we cannot take in because they're too large to pass through the fenestrations. Okay, but now how do we reabsorb this? So in this next region, it's going to be bordered by more blood vessels, and this is where we can have absorption occurring. So our amino acids can get taken back, so can some glucose. So now they are back into circulation. Back into circulation. And this region of squiggly tube that I drew here, this is called the proximal convoluted tubule. Okay, proximal because it is, close to the glomerulus. Convoluted because it has a bit of this convoluted, the squiggly shape, and tubule just means little tube. So here we have some absorption. And now this is going to eventually dip down into the medulla we go. So now to the medulla. So all these structures that we mentioned here, this is all in the cortex. So if you look up here, everything that we mentioned is just in the cortex region. So let's make a note of that. So the arterioles, the glomerulus, Bowman's capsule, I'll abbreviate that to BC, and the proximal convoluted tubule are all in cortex. The cortex. We haven't reached the medulla just yet. Okay, but we will. Now it's down to the medulla. And this next structure, let me draw part of it. Here's the proximal convoluted tubule. Now it's going to dip down into the cortex. And we'll see why it does that in a moment. But then it comes back up eventually. Okay. So this region here, this is the cortex, and below it is the medulla. And this structure here, this loop, is called the loop of Henle. This chunk here, where filtrate travels down, so filtrate traveling down, we call this the descending limb, the loop of Henle. Descending limb. And then this part up here, where filtrate travels up, we call this the ascending limb. Ascending limb of the loop of Henle. Let's first talk about the function for the ascending limb. This part here is not permeable to water. Not permeable to water. So water cannot escape in the ascending limb. It is, however, permeable to ions. Permeable to ions. That means ions can cross through and exit. So let's see here. Some examples we mentioned were potassium, sodium, and we also have chloride ions. Now, if you were to think of these as solutes, substances that can attract water, it's going to be full of these solutes. Thus, this is going to make this interstitium, this environment here, hypertonic. So it's going to make this area, the medulla, make it hypertonic. What this means is we're going to attract water. Attract water. But water can't get through on this side, it's going to get through from the descending side. So now because this environment is so hypertonic, it's going to attract these water molecules, thus allowing us to reabsorb water. So descending limb main function is for water reabsorption. Water reabsorption. Okay, now we're back into the cortex and onto the next squiggly structure. So again, here is the proximal first. Proximal squiggly dips down into the medulla. We come back up. Now we have a second squiggly. We call this the distal convoluted tubule. Distal because it's farther from the glomerulus. Convoluted because again it still has that squiggly shape. And tubule, all that means it's a little tiny tube. So this is the distal convoluted tubule. And here we just have some additional reabsorption. So in this area we have some additional reabsorption based on the environmental needs of our body. Additional reabsorption. So we can take in more things if we need them. After that, this DCT is going to converge with the DCT of other nephrons. So this is the DCT from another nephron. This is also the DCT from another nephron right here, and so forth. From another nephron. And all these are going to converge into the collecting duct. We call this the collecting duct. And here we have final water reabsorption. Final water reabsorption. Because notice we dip back down into the medulla, and the medulla has many solutes. It's solute heavy, and this causes water to exit. And this is how we can do some final water reabsorption. And hormones can change how much water we can take in. We'll discuss that for a different lab.